This DNA structure is a double helix shown by the two strands of nucleic acid around the structure. In these strands, there are 4 types of nitrogenous bases that construct both the sequence of DNA and RNA. This is C, A, G, and T. Now, each of these terms are either purines or pyridamines, and this means that A and G, or adenine and guanine, are pyridamines, while the T and C are purines. Each one of these nucleic acids has a base pairing. These base pairings are represented by the 2 colors in the middle. A and T are base pairings, and C and G are base pairings. In the middle of these base pairings, there are things that are called nucleotides, and those are the building blocks of the DNA, as represented here. When 3 nucleotides line up in a base pairing, that is called a codon. And when you have all of your codons and your sequencing along the 2 sides of the helix, that makes genetic coding. And genetic coding is the information that DNA stores that is replicated all throughout your body. When forming these base pairings, though, there are lines here that represent hydrogen bonds, and these hydrogen bonds also keep the structure together. When DNA replicates, it's separated by the enzyme called a helicase. The helicase goes down the 2 strands and breaks apart the leading strand and the lagging strand. Now, this process is known as semi-conservative because the helicase comes in and breaks these strands apart at one place in one time. As these helicases going up and splitting the 2 strands, things called binding proteins come in, and they keep the 2 strands separated so they don't form back together when the helicase is moved on. As these strands are splitting, an enzyme called DNA polymerase III comes in and encodes the leading strand in old DNA pieces to form one long strand, which is known as new DNA. And with the focus of DNA polymerase III on the leading strand, this lagging strand is forced to also replicate old DNA strands in fragments, and these fragments are called Okazaki. When the Okazaki fragments are being created on the lagging strand, the enzyme called RNA primase comes down and lays down RNA primer on the lagging strand. Now, then after the DNA polymerase III is done with the leading strand, it comes down to the lagging strand and fills in the gaps with new DNA. Now, then, an enzyme called DNA polymerase I comes in and replaces all the RNA primer with DNA. So now there's a bunch of unlinked DNA on the lagging cell. Lastly, an enzyme called DNA ligase comes in and binds all of the Okazaki fragments, new DNA, and now transform DNA from primer all into new DNA to form 2 separate strands. Transcription occurs after replication. The focus of transcription is to produce RNA, but specifically mRNA. And mRNA basically just carries the genetic information to certain DNA to amino acids. Now, to produce mRNA, a RNA primase comes in and binds to a gene sequence, and pulls one of these strands to be used as a template. When making mRNA, a thing called a promoter region has to identify to the RNA polymerase where to start coding this template strand. And as seen here, it picked the beginning of C, so this is going to be the promoter region. And now our mRNA is basically just a template that's being coded to almost exactly replicate this. The only difference is that now instead of T, which is thymine, it's now replaced by U, which is uracil. Once the mRNA is done coding, the mRNA goes to the amino acids to carry the information, and the pulled apart gene strand returns to the other gene strand, and everything reverts back to normal.